Take a few minutes. Yeah, sure. Wow, the Macedonian paper just came out. Yes, I made it. Yeah, for you. Finally. Yeah, yeah. Yeah, Hanqing and Rachel take a lot of time on answering the question. I feel for that paper, it's kind of being very comprehensive already in the first question, and then we were just asking a lot of curiosity questions. So they still did quite a lot of things. Yeah, yeah. I thought that paper was probably needing very little revision, and I was surprised they still take a lot, which is probably means that yeah, initial deadline was just too aggressive. Yeah, like Hanqing it was starting the new position, and the pretty busy with the experiments. Yeah. Yes, everyone is maybe. You find your parking okay? Yeah, yeah. I added to the B parking with one of my friends. So. Okay. Well, yeah. Hello, everyone. Welcome to San Diego Chromatin Club meeting. This is the very first in-person meeting after almost 4 years, 3.5 years of kind of a pause in person. We did have online virtual meeting, remote meeting, which a lot of people from all over the world dial in and listen. And in fact, it was really hard for us to decide, should we continue that online virtual format or in-person meeting? And after a lot of debate among organizers, me including Ann Bingren and Chris Banner is here, as well as Alan Wan, Diana Hargreaves, and Ani Despande from SBP. We finally realized that we still miss this in-person component of the club. When you say there's a club, if you just meet virtually, it felt differently. So we want to give it a try to rekindle this personal touch among colleagues within the San Diego area. So we try to have it attended in person. So I'm glad that you're all here. I hope this is going to beginning of a lot more interactions. So I want to also say a few words. I think that the mission of Chromatin Club was aimed to promote interactions, indeed, among labs, among trainees, among colleagues, and has been quite successful in that regard. In the past, we have had this meeting on a monthly basis, and we have had a lot of participation. So obviously, after 3 years, we have a lot of new faces. Okay, I mean, you probably don't know each other very well, but hopefully with more and more such events, we'll get to know each other better. And now we changed the format such that we'll have trainee talks, 2 on each session on a monthly basis. So I encourage each of and every one of you to sign up and to present your latest and exciting work, ideally unpublished, but if you felt you want to talk about published work, also please do, because I think in this world, it's information heavy. Publishing doesn't mean everyone knows, so bring it to this podium. So the format is such that the first hour is going to be 2 talks. Followed in that, we will have a happy hour. How long have you? What was your last happy hour? I think well, we are missing that. So it's I want to say special thanks to Seth Rubin there, who from Active Motif, who has been a supporter of this Chromatin Club from the very beginning. I would say, what, 6-7 years ago. And has been, this is the very first Chromatin Club in the country, and then Active Motif has now duplicated in many other cities, even in other continents. So it's actually a very successful model. And again, thanks to the sponsorship, you will have drinks afterwards, snacks. So now I want to also mention, I mentioned the organization team, I mentioned the sponsors, and most importantly, everyone who responded to the email and joined this Chromatin Club. Thank you and very welcome. So today we have the special pleasure to have 2 scientists to talk about their unpublished work, I think. But probably one is in press already and are gaining a lot of attention. So the first speaker is Jintian Zhou, who is currently at Salk Institute. He just got his Ph.D. not long ago, and now he's doing is a postdoctoral fellow at Joe Ecker's lab. So Jintian.
All right, so thanks Bing for the super kind introduction, and thanks for having me here. And today I'm super excited to share with you our recent progress using these single-cell multi-omic approaches to study the brain. So the collective goal of neuroscience is to understand how brain works. So this is a very challenging task, given the complexity of the brain, where there are hundreds of millions of neurons in the mouse brain, hundreds of billions of neurons in the human brain, and a similar number of non-neuronal cells, and extremely complex interaction between them through synaptic and non-synaptic connections. So to achieve this goal, people around the world are taking these multi-scale approaches to understand the principle of how brain is organized, from the diversity of cell and cell types and how they're connecting to each other and what cells are associated with what behavior. So this picture shows you the diversity of the cortical neurons, where you can see they can express different neurotransmitters and located in different layers of the cortex and having different morphology and connection patterns. However, all of these cells actually have the same genome, but they can have extremely different functions. So how could that happen, right? So the part of the reason is because between genotype to phenotype, there are those intermediate molecular phenotype, including the epigenome and the transcriptome, which shape this fundamental translation process in biology from genotype to phenotype. So I guess I don't need to talk too much on this content to this audience. So the epigenome includes the different layers of regulation, starting from the DNA methylation as single cytosine all the way to transcription factor binding, nucleosome positioning, histone modification at different levels of 3 genome structure. And what we focus on in our lab is the DNA methylation and the 3 genome structure, where the DNA methylation can help us to pinpoint the potential regulatory elements in the genome, and the 3 genome structure help us to link those regulatory elements to their potential target genes through physical interaction between promoter and enhancers. So in order to study the cell type specificity of the epigenome, we're taking these single cell approaches where we profile the single cell, the epigenome information within each single cell, and then we merge the cells that are similar to each other to identify cell types and generate a pseudobulk epigenome information for each cell type and compare them with each other. So in today's talk, I will show you our recent progress on using these single cell technologies, starting from how we develop those technologies, and give 2 more examples on how we apply them to build a whole mouse brain cell type atlas and also a 3 genome atlas of the human brain, to especially on how we are developing new computational tools to facilitate the analysis of this new type and new scale of the data. So starting from the technology, in the past 6 to 7 years in the Ecker lab, we're developing single-cell methylation and methylation-based multi-omic technologies summarized here, including SNM-seq and this improved version 2 and 3, which is a plate-based method to profile DNA methylation with bisulfite sequencing in single cells. And we also developed this epi-retro-seq, where we use the retrograde virus to label the neurons projecting to a specific brain regions and sequence their DNA methylome of the nuclei. So for each single neuron, we know both its projection target in the brain as well as its epigenome. Also, we developed this SNM3-seq. So by the chromatin conformation capture technology before bisulfite sequencing, in each single cell, we can simultaneously measure the DNA methylome and the 3 genome structures. So to analyze this single cell epigenome data to study their cell type specificity, it usually involves the 2 sections of analysis. One is the cell level analysis, and the other is the genome level analysis. So the cell analysis include the clustering of similar cells to identify cell types, as well as the integration across the different modalities. For example, this means if you have part of your samples profile with single-cell RNA-seq and the other part of the same sample profile with single-cell DNA methylation sequencing, you can computationally infer the match of cells between the cells profile with a different technology. And in this way, for each cell type, you can generate a profile of both its gene expression and the DNA methylation, and this helps us to study the relationship between these molecular modalities. And the genome analysis involves identification of specific genomic elements. For example, the differential methylated regions, which indicate the potential regulatory elements, and also include the identification of chromatin compartment domains and loops on this contact, on these high-C contact maps. So in the first part, I will then show you some examples of how we're using these technologies to study the cell types in the whole mouse brain to exemplify the cell level analysis. So the mouse brain consists of a wide range of different structures responsible for different functions and behaviors. To study the cell type diversity of each of these brain structures, we're doing a manual dissection of the mouse brain into 18 chrono slices. And on each of these slices, we separate these major structures. And this in total, we have here 117 different structures dissected. And we use this the single cell technology to profile each of them separately, which generate a combined data set of more than 300,000 cells. So the other groups across the brain initiative cell census network also generate single cell RNA-seq data, as well as in Bing's lab generating the single cell attack data of millions of cells. So to deal with this large scale data set as a routine, really require the efficient algorithm and computational framework to deal with them. So the solution we come up with is that we developed a tool called OCUS for DNA methylation data analysis, and this is based on an efficient data structure called ZAR to save the large data set into chunks on the disk, and also use the X-array and dust to allow on-disk computing and parallelization. So the key idea here is that you do not need to load this whole data set, large data set, into the memory, but you just load the small chunk of data that you are currently processing. So it allows the methylation, the cell type classification based on the DNA methylation, as well as the integration across different modalities of millions of cells within several hours on a standard workstation. So as you can, I guess you can already appreciate the complexity of the mouse brain by just looking at the t-SNE, where collectively we identified more than 4,000 clusters across the whole mouse brain. And among these major structures, including the isocortex, the hippocampus, the olfactory area, the cerebral nuclei, the amygdala, thalamus, hypothalamus, midbrain, hindbrain, and the cerebellum. So you can see most of this brain structure has the cell types that are located in a quite different space on this t-SNE, suggesting those brain structures are having quite a different cell type composition. And this is also showing that the cell type complexity of those subcortical brainstem regions are much higher compared to the cortical regions where we most of the previous study focused on. So as I mentioned, this OCUS also allows us to do integrative analysis across different modalities and of millions of cells, including the single-cell RNA-seq and attack data, and also by combining them with the Murphy spatial transcriptome data, we're able to locate these cell types that we identified into the spatial context of the mouse brain. And besides that, we also generated the epiretro-seq data, which is the projection neuron data set with the source and target of the projection covers most of these major brain structures. And by mapping this projection neuron single cell data to this unbiased atlas that I described just before, we are able to infer where are those cell type potentially projecting to. So to make a more concrete example of how the power of using this data set to study the brain cell types. Here I'm showing you the neurons in hypothalamus, where this clustering is jointly defined by single-cell RNA-seq and DNA methylation sequencing. So if we map the epiretro projection data onto this the same space, we could see that the projection neurons are enriched in a small proportion of these clusters, but not all of them. And we can further quantify the proportion of the cells in each cluster that are projecting to different targets. For example, we can see for clusters 76, they are primarily projecting to pons and striatum, and the cluster 0 is majorly enriched for neurons projecting to medulla and some of the other targets. So by combining with Murphy data, again, we can look at the location of this projection enriched neuron at a much finer spatial resolution comparing to our brain dissection. For example, you see these clusters that are circled here are primarily located at these subthalamic nuclei and zonar inserter regions in the posterior part of the hypothalamus. As this is stripe structures here shown on a Murphy map. And we have some other examples of the spatial location of the cell types. And also when we're looking at the sagittal slides, we can see the distribution of this projection enriched clusters from anterior to posterior of the hypothalamus, which potentially suggests the topographical projection pattern of these neurons. So I want to briefly mention one of the interesting observation we have from this large-scale atlas, where previously we usually consider the neurons, the cortical neurons that only project within the cortex. So we call this IT neurons. They are totally different cell types as comparing to the neurons that can project out of the cortex to the brainstem, as shown as we call it ET neurons, as shown as the cyan color on the second row. But we also, in this projection data set, we noticed that there is a small population of neurons in our data labeled as visual cortex projecting that are located in this sign cluster, which is supposed to project out of the cortex. So this makes us hypothesize that this is the population of neurons that can make projections to both the visual cortex and the brainstem. So we do some anatomical experiment to validate this by labeling the neurons projecting from the retrosplenial cortex to the primary visual cortex. And indeed, we see their axon target located in the brainstem regions, including the clypeus and pons. So this validates the existence of this neuron that is suggested by our molecular data. And as a summary of this part, so I've shown you that OCUs can be used for large-scale DNA methylation data analysis and the integration with other modalities. And with this, we can build the integrative cell type atlas across the whole mouse brain. And also by further integration with the Murphy data and the projection data, we're able to infer where the cell types we identified are spatially located in the brain and where they are connecting to in the brain. So maybe these slides, I want to just briefly show you from maybe a big picture view of the evolving of this field using single cell sequencing to study the complexity of the brain. We're starting from 2015 to 16, people starting from thousands of cells and dozens of clusters, and all the way to nowadays, where we're looking at millions of cells and the thousands of clusters in the brain. And this really provides a huge resource for us to understand this the part list of the complex brain systems. And in the next part, I'm going to show you how we study the cell type specific 3 genome structures in the human brain to show the genomic analysis. So again, start from some challenges in this field at the beginning, where the first thing is about the data sparsity. So if you're looking at the bulk Hi-C contact map, they are usually generated from millions of cells and billions of reads. So you see this dense contact map, but when you're looking at the single cell Hi-C data, they're usually just having a few 100,000 contacts per cell, which make this matrix super sparse and very hard for the downstream analysis. And also, I want to briefly mention the intrinsic heterogeneity, which means even within the same cell type, the different cells are quite variable in terms of its DNA structure by nature. So, and how this variability within a cell type across cell are comparing to the difference of genome structure across cell type are not well understood. So to really address these questions and to analyze the single cell chromatin structure data, what we developed is a package called SC high cluster to impute this sparse contact matrix through first step, a linear convolution step that borrow information from the neighbors on the linear genome, and a second random walk step that borrow information from its neighbors in 3 dimensions. And these 2 steps of imputation makes the matrix much denser and easier for the downstream task. So with SC high cluster, we can do single cell embedding to identify the cluster, the cell types at a reasonable resolution, as well as identify the topological associated domains within single cells. And also by optimizing existing tool called Snap Hi-C, we're able to identify chromatin loops with as low as just 100 cells, which used to need more than 1,000 cells. So equipped with SM3CC and SC high cluster, we profiled the cells from the adult mouse brain for 17 different dissector regions. And we also profiled the developing human brain from mid-gestation all the way to the adulthood. And we also have other atlas of the 3 genome structure for the whole mouse brain, as well as for a coarser atlas for 15 different adult tissues, which I will not have time to talk about today. So today I will focus on these 2 human brain data sets, where still by looking at the t-SNE using either the DNA methylation or 3 genome structure as the input, you can already see the cell type specificity of them, which is indicated by the separation of different colors representing the different cell types. So this is important because most of the cell type specificity analysis in previous studies are of the 3 genome structure are focused on different cell lines or bulk tissues. And this is really the first time that we're pushing this, this study into such a fine-grained cell type resolution. So by coloring them differently, we can also see the specificity of the genome structure across the different brain regions, as well as across the developmental trajectory from second trimester, third trimester to infant and adulthood. So as a more concrete example, I'm showing you the contact map at from the left, the SOX11 gene, which is a developmental gene, and you see its genome structure, the domains at the flanking region of this gene has decreased the strength as it's going through development. In contrast, another gene, the neurexin gene, have its gene body domain getting stronger and stronger during development in the excitatory neurons. So next, we look at the cell type specificity of chromatin loop and domains. The way we're doing it is we are using the interaction strengths at the loop anchors and the single cell domains we identified as input to generate a cell embedding and classify the cell types. As you can see from this embedding and the quantification, the chromatin loops has a better power to distinguish the cell type comparing to the domains, which suggests that they are more specific across cell types. So next, we also look at the relationship between the chromatin interaction and DNA methylation. We see that there's a very strong anti-correlation at the loop regions between the chromatin between its interaction strengths versus the DNA methylation in the adult human brain across the cell types. However, if we look at the developmental data and look at the same thing, basically we start to see this discrepancy, where on the left, you can see the major switch of chromatin interactions through development happens in the middle of somewhere of the second trimester. However, the major switch of the DNA methylation of the genes anchored at these loops are occurring in the middle of the late gestation and the infant stage, which suggests that in this example, in the astrocyte, the chromatin are changing ahead of the DNA methylation. So we also look at this discrepancy by using these two different modalities to separate different cell types. So as shown in the previous slides, in most of the cases, DNA methylation is better to distinguish these different cell types comparing to 3 genome. However, in this case, where we're trying to look at the two subtypes of radioglia cells in this mid-gestation, in very earlier stage of development, you see only the chromatin structure can separate these two cell types, but not DNA methylation. And when we're further looking at what leads to this separation of the cell type, we see the chromatin structure at this SALB2 locus, which is a marker gene of adult excitatory neurons. And we see in the radioglia 1 subtype, the chromatin structure is more similar to the adult excitatory neuron. And in the radioglia 2 subtype, the structure is more similar to the adult oligodendrocyte, which suggests these two subpopulation of the radioglia potentially serve as progenitor cells of neuron versus non-neuron. And we further model this discrepancy between DNA methylation and 3 genome changes. Using this analysis, we call it chromatin potential analysis, where for a single cell, when we're looking at its 3C profile, we're trying to find the most similar methylation profile across all the cell. And then we realize that this methylation profile usually corresponding to a cell that is in a later developmental stages comparing to a 3C cell. So this again suggests that in this system, we're seeing this time discrepancy of development, and the 3 genome structure usually changes ahead of DNA methylation. And I will just skip this. And as a summary, today I'm showing you the technology we developed, including SNM-C-seq for DNA methylation profile, SM3C-seq for jointing profile DNA methylation and genome structure, and epiretro-seq for DNA methylation and neural projection study, and the computational tools, OCUs for methylation and integration analysis, and as a high cluster for genome structure study for cell type clustering, as well as identifying the chromatin domain loops. So with this technology and computational tools, we build a whole mouse brain atlas to show the complexity of cell types in the brain. And we build a joint atlas of DNA methylation, open chromatin, and gene expression. And also by linking it with MERFISH and the epiretro-seq data, we're able to study the projection and spatial location of those cell types. And for the 3D genome atlas, I've been showing you the cell type specificity of 3 genome structure, which is kind of underappreciated in previous study. And I'm showing you the correlation of methylation and genome structure across the cell type in the IDOT, and also the temporal discrepancy between them along the development. So I want to definitely thank everyone in the Ecker lab, and also a huge team of collaboration without whom this large scale work can never be achieved. And at last, I want to do a advertisement. So I'm going to start my own lab at the ARC Institute in Palo Alto, which is a new institute running in partnership with Stanford, UCSF, and Berkeley. If you are interested in those single cell genomics and this new technology and computational tools, if you know someone who is interested in that, definitely don't hesitate to contact me. And I'm looking forward to work together with you. And we're actually hiring research assistants and postdocs. So thank you for listening, and happy to take any questions.
Yeah, we have time for a few minutes for questions. Maybe I can start by asking about this temporal difference between methylation and the 3C. If I understood correctly, you said 3-dimensional chromatin structure changes occur prior to the methylation changes. Do you have some example of how this occurs and the explanation of why?
Yeah, that's a great question. So here, basically, I'm showing you several examples of this this discrepancy of timing. Like this is showing the very early stage of the development where the 3 genome structure has already distinguished those types, but not for DNA methylation. But still, regarding for the mechanism of why this is happening, this definitely need more of this study. So there are existing study showing the relationship between the how DNA methylation and the 3 genome structure could be co-regulated. For example, the effect of DNA methylation on CCF binding, and also some transcription factor could together, like for example, FOXP1 can regulate both the genome structure and the DNA methylation during brain development. But however, at this resolution of specific time point, I think we still need more both modeling approaches and the experimental approaches to validate what is really happening here. Yeah, and also I want to mention for this time discrepancy, I'm kind of showing you some example which show more of a clear pattern, but also we recognize this is a global pattern, but for specific genes, there could be a different story. So if you are looking at a gene that you're interested, whether like which modality is changing first, probably need to dig into that gene to really look at it.
I was wondering if those colors here are 5 the number of cells in this group. I was wondering how do you navigate the group for specifically one cell type?
Yeah. Yeah, that's a great question. And also, I think here you are right. So all of this loop calling algorithm is biased because of the coverage, and it's really hard to normalize it. But this you can, but this is also intuitive, right? Because when you have more reads, this is more confident that you can identify those loops. But in terms of here, for quantitative comparison between cell types, we are not directly doing intersect of those loop list. However, we are looking at the contact maps and doing statistical tests of the real values rather than just rely on the 0, 1 value of whether it is a loop or not. So in SDI cluster, we've in the past months, we've updated the documentation and to include all the scripts, I think, that we're using for the human brain atlas analysis. So that includes, if that's helpful, yeah, definitely I will recommend like reading this and to implement to your own data. And if you realize any bugs and something that doesn't work, just tell us. Yeah. Yeah.
Have you seen like in the fact the problem performer or the like the comparing the people who use brand to like add or brand to stuff different.
Yeah, exactly. Thanks for catching that. Yeah, I didn't mention the distinction between non-CG methylation versus CG methylation in this talk. And yeah, for the other audience, it's like there are two types of methylation based on the sequence contact. The traditional, like we're looking at the methylation happen in the CpG contact, where it's happening in almost all the mammalian cells and cell types. And the methylation level through development is the global level is staying similarly across the development, but there are some regulatory elements has their methylation changes. And in contrast, the CH methylation has an accumulation process where in the early stage of development, you almost cannot detect any CH methylation, but it's accumulated through development during the second trimester to infant stage. And that start to show the cell type specificity correlated with gene expression. And going back to your question, for clustering the using methylation data, we are using both CH and CG methylation. And in this case, definitely an earlier time point, the CG methylation is better in terms of resolving the cell types because the CH are still quite noisy at the second trimester. But it starts to work gradually when you're going through this development process. Yeah, so my answer to this is we are trying to use the CG methylation and we cannot separate either. But yeah, I realized I'm not saying there's no DMRs between these two populations. So maybe there are too few number of DMRs that not able to separate them, or the major dimension of the variation is not corresponding to this separation. Yeah, but yeah, this is related to the algorithm, but I would say with the framework that we routinely use, the CG methylation is not doing a good job here.
Thank you.
Yeah, I will move in January. Yeah. Let's talk more later.
So just a brief introduction. Kai is currently a postdoctoral fellow in my lab. He's done a great job. Now it's also been started his lab